Hello, welcome to another episode of Science Talks, a conversation hosted by the University of Arizona's BIO5 Institute. My name is Amy Randall Barber. Today I have Caroline Mosley co-hosting with me. Caroline is the Senior Manager for Communications and Content here at BIO5. It works very closely with the Postdoctoral Fellowship, which we will hear more about in a little bit. Caroline, thank you so much for joining me today in hosting.
Great. Thank you, Amy. It's great to be here. It's very fun to host interviews with our BIO5 Postdoctoral Fellows. I'm excited to talk more with our guests today and learn about volatile organic compounds and how they contribute to air pollution. Air pollution, which has implications for human health, particularly in urban areas, may be attributed to volatile organic compounds, or VOCs. VOCs in the atmosphere have various sources, but of rising concern are those originating from personal care items, like cleaning products and industrial solvents. Today, we are joined by Dr. Gemma Purser, a 2024 BIO5 Postdoctoral Fellow in Dr. Laura Meredith's lab in the School of Natural Resources and the Environment at the University of Arizona College of Agriculture, Life, and Environmental Sciences. Specializing in atmospheric and analytical chemistry, Dr. Purser works to further understand VOCs so we better understand how they impact urban green spaces and air quality. Gemma, thank you for joining us today.
Yeah, thank you for having me. It's exciting to be on the podcast. So, yeah.
Wonderful. Well, we like to start off every podcast the same with a few fun rapid fire questions that are meant to be just simple and easy to answer. So what is your all-time favorite movie?
Okay, so I've been thinking about this, and I think it's got to be like a James Bond movie, maybe one of them, Skyfall, something like that. Just being from the UK, so.
Solid choice. Yeah. If you could live anywhere in the world, where would it be?
So I've thought about this too, and I guess I've been lucky and fortunate to go to a lot of different countries through doing research and science, but I guess there's nowhere kind of like home. So yeah, back home.
Okay.
Would be ultimately, yeah. So
Okay.
UK. Mm-hmm.
Nice.
If you were stranded on a desert island, what 3 things would you want to have with you?
Yeah, so there's lots of practical things that I guess you'd probably need, but I think I'd probably need tea, because that's essential, and everything can be solved with a cup of tea, any problem. So being stranded on a desert island would probably feel pretty good too. And then I guess, I don't know, my binoculars, so I could do a bit of bird watching, maybe useful for looking for ships and things like that too. So yeah, so maybe that's only 2, but okay, tea and biscuits. Yeah, there you go.
Tea, biscuits, binoculars.
Yeah.
Perfect.
Love it. So we always like to get to know our guests' background and how they got into science. Can you tell us kind of how you got into science and it became an interest for you?
Yeah, sure. So as a kid, my dad was really into nature, and so I often used to go out walking with him, and he'd teach me things about different plants or bird species and things like that. So that's really where my passion and interest came from for the natural world. And then at school, and I guess what you guys call high school, what we call secondary school, I did chemistry and biology there, and I really kind of thought, yeah, this is really what I want to pursue. I liked all the questions that would come up and kind of being observant about the natural world and trying to answer those questions. So, and then from there, I took my A-levels, which I don't know what that equivalent is in the US, but and then I went to university where I studied chemistry and biology, the University of Derby. And then actually I decided I wanted to get a job. So I worked in a pharmaceutical company. I also worked in a dairy testing milk, so I've done lots of different jobs. And then whilst I was doing the pharmaceutical kind of role as an analytical chemist, I decided that I wanted to do a master's. So I did a part-time master's, which took me 4 years, and I did that alongside my 9 to 5. And then from there, I decided maybe that, I really wanted to do something a little bit more with my science. So I worked for the British Geological Survey, doing geochemistry. And this is really where my passion for scientific research took off. It kind of opened my eyes to this totally different way of working, having these, working on the cutting edge, having questions that you have hypotheses and testing them. So yeah, that was really exciting. So yeah, that's kind of my journey. And then after 8 years working at the British Geological Survey, I thought rocks, so I worked on things like understanding the chemical changes in rocks when we inject carbon dioxide underground to try and store carbon dioxide there. So, and also things like what happens if you, the chemical reactions in a radioactive waste storage repository. So totally different to what I do now. And after a while, I realised rocks are pretty slow. Some of the experiments, we put on would take somewhere between 5 years, or even some of the oldest experiments were 25 years long. And I wanted to work maybe in a field of science that was much quicker. So I decided that I would go and do my PhD. And this time, really focusing on trees again. So my original passion. So that's kind of been my journey so far.
Wonderful. Thank you so much for sharing all that with us. That's really cool that, you can root it all the way back to secondary school. So you did your PhD in Scotland. Can you tell us about your experience in Scotland? Had you been to Scotland prior to that?
Scotland was great. So it is a separate country from England, where I'm from. And so my time in Edinburgh was great. I as you say, I studied at the University of Edinburgh, and I also worked in collaboration with 2 research organisations in the UK. One was called the UK Centre for Ecology and Hydrology, and the other one was Forest Research. And they're very much focused on environmental change and forestry. So for my PhD, I really wanted to understand the impact of tree planting in the UK, like in terms of if the UK, for example, could grow its own source of biomass for bioenergy. So typically, actually, about 11% of the UK's electricity and 6.4% of the UK's heating energy comes from burning biomass in 2022. However, most of that biomass actually comes from the US and Canada. So really, to make it more sustainable, we need to grow our own biomass. So my actual PhD project was focusing on understanding what other impacts are there of growing many more trees in the UK. And so, the good part of this is that we can grow trees, and we know that trees are a sink for carbon dioxide. They take up carbon dioxide, they give off oxygen, and so we can use that stored carbon, and then we can burn that to make energy. So that's kind of this, in theory, more greener way of producing energy. So we know that growing trees in the right place can act as a sink for carbon dioxide, and they give oxygen out, but they also emit some other compounds in much smaller amounts called volatile organic compounds. And there are about 1,700 different species identified, and the plants can emit them and trees can emit them from their leaves, roots, flowers, their fruits and things like that. So for my PhD, I was really interested in measuring the emissions of these compounds, and particularly a group of compounds called terpenoids, and how much of these are being emitted from different types of tree species and that we could grow for bioenergy. And like I say, they're important because although they're only emitted in relatively small amounts, they're very reactive in the atmosphere. And so especially in the presence of things like nitrogen dioxide, and typically we find high levels of nitrogen dioxide, things like that in urban environments. And so nitrogen dioxide can react with these volatile organic compounds, these VOCs as we'll call them now, and create ozone. And ozone, although we've heard about ozone being really important in the stratosphere, like the really high levels of the atmosphere for helping us protect us from UV rays from the sun, in the lower part, in the troposphere, the layer of the atmosphere where we breathe and where we live, it's a problem because ozone can be an air pollutant and it can cause respiratory problems for humans, it can damage crops and things like that. So it's really important to understand what these VOCs are being emitted and how they might be involved in these reactions in the future. And what we found through that work that I did for my PhD is actually, yes, if you plant the trees that emit much more VOCs, then you do get a small increase in ozone in certain areas of the UK. But actually, the trees were a really great sink and a really benefit to a different source of air pollution, which is called particulate matter. So what would happen is the particulate matter in the atmosphere would deposit onto the leaf. So they would get trapped by the leaf surfaces and be removed from the atmosphere. So actually, trees are planting more trees is beneficial for the atmosphere and air quality, maybe then, which outweighs some of these other processes that might occur from planting trees. So yeah.
So there's all different kinds of trees. So I'm going to assume that there has to be some differences between trees, or is the uptake? Are you looking at this in terms of kind of like more of an ecosystem perspective, and then looking at individual trees and certain species, or are you trying to kind of understand it as a whole right now, like the whole dynamic of kind of like trees and forests?
Yeah.
These particularly matter.
Yeah. So so for my PhD, I was really at first like wanting to understand the particular tree species, how much they were being emitted. But yeah, then what you do is you can use that information then to put into much more bigger models, larger models, where we can understand how air chemistry might behave and things like that on an ecosystem scale. And I guess one of the things that really struck me during my PhD was that I was really focusing on the leaves and the branches of the trees, but what happens to the things like in the soil or the forest floor? So how are they contributing to these processes? How do they contribute to VOC emissions, or like say, how can they maybe remove VOCs? Yeah.
Okay. That's really interesting, yeah, because I know that you have to start at kind of the micro level, but then go out to the macro level, and connecting the two can always be challenging. So, so we talked about your science, but you're also a researcher who's really interested in science communication. Can you tell us about I think it's called iLeap Scientific Steering Committee, and kind of how that's impacted your scientific perspective?
Yeah. I'm super passionate about science communication and bringing together like research, like the research community in my own field, and also more widely connecting different people from across the globe and in different areas of research. And iLeaps, which this is a bit of a mouthful, but it stands for Integrated Land Ecosystem Atmospheres Processes Study, and it's a research organisation that focuses around building opportunities to bring researchers working on different aspects of science related to the biosphere and the atmosphere, and how they might interact, and bringing those people together to collaborate in the future. So I'm an early career member of that scientific steering committee, and it's been an invaluable experience for me to be able to meet and connect with many new people through being part of the committee. So it's not only brought benefits to my own research and my own career, but also I've really tried through that work to create events and resources to help build our community. So actually I have my own iLeaps podcast, which it's quite different for me today to be here on the other side of a podcast. I'm normally the one doing the interview, the questions.
What's your podcast called?
It's the iLeaps podcast.
That's the name. Okay. That's cool.
So it's on Spotify, so you can check that out. And through that, I've interviewed many researchers from Latin America, Africa, Europe, and I've still actually got some podcasts to release from Australia and New Zealand. But like things like Twitter poster competitions as well, where you'd have like 24 hours of crazy tweeting where early careers researchers could share their posters and their work online and get feedback from senior scientists. So it's been really fun. Yeah, it's been really cool.
Yeah, I agree. Science communication and collaboration is invaluable, and it's all about kind of finding the people to talk to and kind of make those connections, which can be challenging, but we do have a lot of platforms to do it now, even.
That's true.
You can pick your favorites.
LinkedIn is the new one, right?
Yeah, right, exactly. Yeah. Now and then you have to build a whole new audience. But we will definitely give a plug for your podcast as well, so people can listen to that.
Thank you.
So what was it that brought you to the University of Arizona and to BIO5?
Okay, so this is another story. There's many stories. So during my PhD in 2019, I took a 3-month break, and I came to the US to volunteer as a researcher on a project that was not directly related to my PhD. And through a colleague at the UK Centre for Ecology and Hydrology, I was told about this large project happening at Biosphere 2. And that they were this really cool experiment where they was going to label the atmosphere with this carbon-13, this isotopic tracer, where they could track all the carbon and what was happening in the rainforest biome within Biosphere 2. So I actually came and joined the experiment. The experiment was called B2 World, and it was led by Professor Christiana Werner from the University of Freiburg, and also Professor Laura Meredith here at the University of Arizona. And it was really during this project that I first came to Arizona and Tucson. I got to come out and hang out in the desert, which is really cool, up at Biosphere too. And that's where I met Professor Meredith, and I saw her lab group works on really cool things. So she works on ecosystem genomics, linking soil microbial activity to processes with different trace gases, so things like hydrogen, but also VOCs as well that we've talked about. And I really love her work. And it's kind of, she has a lot of questions that she's working on that kind of are aligned with my own kind of ideas too. So and she's got a wealth of knowledge. And it's not, not just in just Dr. Meredith's lab, but also in the wider BIO5. There's many different researchers, great facilities, core facilities here, that provide the right scientific tools and methods to be able to do my type of science.
And we did not pay you to say
no,
that.
No, but
I was going to say, thank you for plugging our research course. They are amazing.
So much for the honest testimonial. I mean, really. Yeah, that's wonderful.
Yeah, we're always trying to tell people about the different services
All the
and
cool
tools
services,
and the staff that are here to help. So that's great. That's great to hear that. That's great to hear that it's really aligned with your goals. And that's wonderful.
Thank you. So you mentioned being a part of Dr. Meredith's lab. Do you want to talk a little bit more about being a part of her lab and just kind of what you do for her lab?
Yeah, sure. So I moved to Tucson almost a year ago now to work with Professor Meredith as a postdoc on VOCs and particularly their transport and fate within soils underground. So as we discussed earlier, VOCs can be emitted by trees and things and plants from their roots. So smaller compounds, for example. And when we say smaller compounds, we kind of like maybe define them in terms of how many carbons they have in their structure. So small compounds might be things like methanol or acetone, or larger compounds, they might only have 1 or 2 carbons in their structure. And then larger compounds such as terpenoids, such as isoprene or pinene or caryophyllene. And I just want to make this point here for your listeners that these are the things that maybe when you have a massage and you have or put a nice candle on that smells nice, or you walk through a forest and you can smell all these wonderful smells. These are the compounds that we're talking about, which are known as terpenoids.
Thank you.
Yeah, no problem. And these are much bigger molecules, and they might have 5, 10, or even 15 carbons in their structure. So the size of the molecule and the type of molecule might have an impact on the way that it moves through the soil. So it could maybe get stuck on different surfaces within the soil. It might dissolve more easily into the water within soils. And some of these compounds might be easier or more difficult for the microbes and the bacteria within the soil to eat and consume and use for their own sources of fuel. So I really wanted to understand these processes that occur in the soil. And for this work as well, I must add that I've been working in collaboration with Iridian Research, who are over in Boston in Massachusetts. And the reason I work with those is because they have a state-of-the-art mass spectrometer. And to break it, we call it the VOCs. And to break it down, it's called a proton reaction transfer, which just basically means they use water, and we have to ionize the compounds of interest, our VOCs, so they have a charge on them. And then at time of flight, that just means that the different size molecules and different masses move down a tube within the machine at different rates, and the heavier molecules take longer, and the smaller molecules are much faster to move through this tube. And then the other part is the mass spectrometer. So we can separate all these different compounds by how much they weigh and how big they are, and also their charge. So this instrument allows us to see really tiny trace amounts. So when we're talking trace amounts, we're talking like parts per trillion in the soils. And also Iridian, and along with Dr. Meredith, have produced some probes that we can put in the soils. And we can use these probes to sample that air within those tiny air spaces within the soil too. So I've really been interested in understanding how natural VOCs are behaving in soils, and that's why I've come to Professor Meredith's lab. Yeah.
Awesome. Well, and then that kind of segues right into the next question, which is, how does her lab align with your career goals?
Yeah, so, the work that she does is really inspiring. So I've learned a lot here while I've been here. And I've also learned a lot from the rest of my colleagues within the lab as well. They've all got their own specialities, which I can learn from. And it's been great to help me develop, as a scientist in this postdoctoral phase in my career. And ultimately, I guess, it's helped me to be able to apply, science is much reliant on funding. So it's helped me, these experiences have allowed me to apply for my own research grants and to answer more of the questions that interest me. And ultimately, I'd like it to transition into where I can have a longer fellowship role or, my own permanent academic role in the future. So yeah, it's been a really great way to start trying to understand what is my niche within this field of VOC science.
Wonderful.
So thank you for sharing all that about your research. If you kind of had to give, there's so many different pieces to your work. Is there a short summary you would give in terms of is there a takeaway you'd want kind of a lay person to understand about your work?
So I guess I really work in this area of volatile organic compounds, and that's the thing that interests me most. And these volatile organic compounds can come from natural sources, which I'm really interested in understanding, but also they can come from anthropogenic sources, which anthropogenic just means man-made. And some of these VOCs are naturally there, and that's fine, but some of them are there because we put them there. And I guess my work is trying to understand what impacts these have and trying to separate those impacts. And some are beneficial reactions and some are not so good. So
yes, it's quite gray area that you have to tease apart.
Yeah.
And then can you just tell us a little bit, you kind of mentioned mass spectrometry and probes. Can you just talk about kind of the instruments and the tools you use to do the research that you do?
Yeah, so we have different tools that allow us to study volatile organic compounds. One I've already talked about, this PTR TOF mass spectrometer. But then also we have other things we can use. We can have different chambers where we grow, we can have soils or we can grow plants, and we can look at the emissions given off or taken up, VOC emissions given off or taken up by the soils or plants, for example. We also have all the microbial tools, like particularly what we have in BIO5, where we can look at what different types of bacteria or fungi are in the soil. What we use their DNA or their RNA, their activity, their genes to understand how are they using these VOCs in the soil, and who is there. Like, what different types are there? Yeah, so we have a lot of these different tools as well to look at microbial processes. So things like we can look at their DNA, so we can find out which microbes or fungi are in the soils. And we can also look at their RNA. We can understand their activity and what genes they might have to able to consume these or take up these volatile organic compounds, or what genes or activities might be related to emitting these VOCs from the soil. So yeah, they're the different tools we have.
Do you have one that you think is more challenging than the other?
I mean, for me,
for you personally,
this whole microbial community, DNA, RNA, genes. This is all new to me. This is not my field. I'm very much come from the atmospheric chemistry side. I'm used to seeing measuring things on the sort of analytical instruments. That's very much my kind of comfort zone. Whereas, so I'm really interested and excited to be able to learn more about the other tools. Yeah.
That's why it's great to have good collaborators that you can lean on to help you learn new things. So that's great. So I also want to mention you're a 2024 BIO5 Postdoctoral Fellow, and you've talked about your project a little bit with Boston University and the Aerodyn research. Could you just tell us a little bit more, what you hope to accomplish with that?
Yeah, so I guess what I want to make clear in this podcast is, when I talked earlier about the work that I did for my PhD, that was very much looking at natural VOCs emitted by plants. But my work for the BIO5 Fellowship is really looking at anthropogenic sources, like we said earlier, man-made sources. So volatile organic compounds are in cities and in the air, in cities in particular, because they're emitted from different sources such as like paints or pesticides, or even the things that we use to shower with, or when we put that scented candle on, or we clean our clothes. They're all VOCs that get emitted into the atmosphere, and our homes are a big source of VOCs. And actually what's really interesting, a lot of studies are finding now, is that we've made many efforts, for example, to reduce industrial sources of VOCs through legislation and things like that, whereas our homes are still pretty much unlegislated. And there's not, and actually they're becoming, the VOCs or from kind of personal care products are becoming the more dominant VOC source within cities. So I was really interested in understanding then, so what is the role of urban green spaces? How can the soils in cities, in our parks, and things like that help to draw down some of these VOCs in the atmosphere? Because as we've said earlier, we know that VOCs can react with other nitrogen dioxide and other compounds like that to create air pollutants, and that has an impact on air quality in our cities. So what is the role of urban green spaces, and how can soils help, and in particular those microbes in the soil? Yeah.
It's always those interfaces where there's a lot of questions.
Yeah.
So, and then I saw on Twitter actually that you were recently awarded a grant. Can you tell us a little bit about that?
Oh yes, that was quite exciting.
Yeah.
So that's like kind of the first grant, the bigger grant that I've won. So this was actually an internal grant through the core facilities here at BIO5. It's really, so through that we can start understanding, now we've won the grant, we can start doing the work to understand how the soil has a memory, basically. So how long do these effects where microbes take up these VOCs in the soil, how long are those effects present? How long do those communities stay active for when they don't have a source of VOC? So it's kind of really understanding like their behaviour in terms of like, if you think of a soil having a memory for taking up those VOCs, how long does that last? So it's going to be exciting.
So is that working with the core facility, like using the instrumentation through the core facilities? Yeah. Okay.
Yeah, so the instruments through the core facilities, yeah.
That's
the
super
genomes.
exciting. Well, great. We're glad we're using the BIO5 resources. That's awesome. That's so exciting to hear.
So you're also part of the Center for the Integration of Research, Teaching, and Learning program. Can you tell us what this is and how it aligns with the work that you do now?
Yeah, so this is CERTL, and this is a really cool thing for postdocs here in the University of Arizona. And it's really about trying to give us the skills and arm us with the tools that we can use in the future to teach undergraduates, which kind of learning many different techniques to really engage undergraduate students, and we do different activities. So there's a co-teaching element to this, where I shadow and work with one of the faculty here at BIO5 to give different lessons on a course. But then also much wider, there's many activities. Like I recently just took part in an online activity where I was using art to express science. So it's really varied different things, activities around that, and it's a really great thing to be part of. Yeah.
We're very into the art and science thing here at BIO5. We have a few different collections here. I don't know if you've seen them hanging in our building, but I encourage you to walk around and take a look.
And we just had a new one installed, so there'll be a story about that soon.
Oh,
so
great.
it was a scientist who was very passionate about art. So all of his art is like one is like about ozone. The one in your office is like something with a jungle.
The jungles, yeah.
Something with carbon and jungle. So anyway,
yeah, but it's like another great, like we say, like it's another great communication tool, right?
Yeah.
So some people, sometimes it's better to express things in visual elements rather than either words or written statements. So
I agree. Yeah,
absolutely. So what are you most excited for about your future?
This is a big question.
I know. It's very open-ended.
Yes. Answer whatever you like.
I guess seeing where this research can go. It's like ultimately, like science for me is pretty exciting. I mean, don't get me wrong. There are lows sometimes. Like let's put that out there. We've got to be real, right? But there's the excitement is in having a theory and then or testing the hypotheses and it kind of coming true, or it's those also those surprises along the way that you didn't even know you was going to discover. So I guess my future, I'd love to keep working with these particular types of compounds and understanding more about them in our ecosystems and in our world. So that's kind of where the future. That's what I'm probably most excited about. Just carrying on doing the science that I love. Yeah.
Cool.
That's great.
Do you have any exciting moments in your scientific career that has happened like super recently? The grant being one of them, I would imagine.
And I also got an award. For a poster award.
Nice.
At the.
Is it at the College of? Well, it's like the College of Agriculture, Life and Environmental Sciences.
You. Yes. There's all these different acronyms as well.
I know. I appreciate you spelling them all out because I'll be googling them later.
Yeah.
So that's great.
So that was pretty good.
Was it what kind of award was it?
It was a poster award. So and so it's always nice to be recognise that you've done some useful work and you communicated it well. So so that was exciting.
That's cool. Yeah. Was it like a graduate poster session?
It was like it was a showcase. So so in that college that everyone could show their work, most recent work. So
awesome. Yeah. Well, congratulations.
Thanks.
Yeah. So this is another kind of big question, but do you have a mentor that really impacted your life?
Yeah, I've been thinking about this one, and I guess I've not had one single mentor. I mean, some people are fortunate to kind of work with somebody their whole career, but you've seen from earlier that I've had a very varied career, and it's not been very linear. I've worked in many different fields within science. So I really would say that I was kind of what they say, like raised by the village, right, or the community in terms of science, and that's why I guess I'm so passionate as well about making sure that there's opportunities for other people to have that same input from the community and creating those opportunities. I would say I've drawn on lots of different people. It's always good to have champions, right? And like Dr. Meredith and my PhD supervisors are very much champions, and even all the great scientists that I've met through iLeaps as well, from many different countries, have also kind of been invaluable in my career. So yeah.
Yeah. Great answer. I think we hear that a lot on the podcast is, it's a chance. A lot of people give shout outs to a lot of different people, but no one can say, there's just one person. I agree. It does take a community. So, so what is your why?
I don't know what this question is.
Another open-ended question. I think we like to kind of dig into just our researchers and kind of why you do what you do. You gave us like a long, you gave us your backstory, but what is your why? Yeah.
Why? Like, how can you not ask why? I would say. Yeah. You observe things every day, and it's important to understand why. When you make these observations, why are they happening? Why do they work the way they work, and how? And all the other questions like that. So I guess it's just intrigue, isn't it? It's just like it's an exciting mystery to be solved. Something like that.
I agree. There are a lot of questions out there in our world that we don't understand. So we need people to ask all the whys and why not. So wonderful.
Wonderful. Well, that pretty much wraps up all of our questions today. Thank you so much for joining us today. It was great talking to you.
Okay, thank you. No, it's been really fun. Thanks very much.
Thanks.
Absolutely.
Thanks to our listeners for tuning in to another episode of Science Talks. Continue the conversation with us next time as we learn more about the amazing science happening at the University of Arizona's BIO5 Institute.